अस्सलाम वालेकुम स्टूडेंट्स। उम्मीद है कि आप लोग ठीक होंगे एंड विल बी एंजॉइंग द कोरोना सीज़न। मींस कि छुट्टियां आपकी बहुत अच्छी गुजर रही होंगी। आज हम पढ़ेंगे फ्रॉम द स्टार्ट ऑफ द जेनेटिक्स, व्हाट इज द basics of the genetic, and what is the history of the genetics, and what was the reason behind it. What are the research work of different scientists that lead towards the birth of the genetics. The history of genetics seems to be as older as the birth of the human being, but there is a very scarce no literature is actually available from where we can get the idea as where the birth of the genetics is occurred. But mostly people thought about that the field of genetics is start from the era of the Mendel, or we can say the after the Mendelian experiment we can foresee that the field of genetics is started. If we look on the Greek philosophers, the Hippocrates and the Aristotle obviously thought extensively about this fact and developed theories to explain resemblance among the relatives. Hippocrates and Aristotle worked a lot to find about the resemblance between the offsprings with their fathers and mothers. The first two names, which we can say the first two names that can be you can say that can be called uh, uh, the first two scientists who work on the genetics are we can say those two are the Hippocrates and Aristotle. 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 Uh, but uh, their findings, but their theories are not in accordance with the current, or we can say, are not in accordance with the modern genetics. They can't explain uh, correctly about the genetics. By the synopsis of their ideas, the reader is referred to the history of genetics by the H. Stubbe. Genetics as we know it today, based on the gene theory of inheritance, which begin with the work of the G. J. Mendel. Mendel is appropriately called the father of genetics. His precedent-setting experiment with the garden pea plant, the Pisum stivum, the scientific name of the plant is the Pisum stivum, published in 1866. Uh, it cannot get the attention of the different scholars at that time. The conclusions that he draw from his elegant investigation constitute the foundation of today's science of genetics. There are many questions regarding the success of the genetics. The major question, why was Mendel so successful in discovering basic principles of genetics? Why the Mendel was successful? The basic question on the Mendelian genetics, or on the Mendelian discovery regarding genetics are the why uh, he was successful, why he was successful. In the later chapter, we will get the answer for all this, all his success. Mendel was not actually the first to perform the hybridization experiment, but what was the reason behind this? He was one of the first to consider the result in terms of the single trait. Most of the people at that time, most of the scientists at that time were considering the complete organism for the study of the genetics. Segret in 1826 had studied the inheritance of contrasting traits. The next scientist is the scientist before the Mendel was Segret. Sageret was in the 1826. He studied inheritance of contrasting traits. Other of the Mendel's predecessors had considered whole organisms. I told you that Mendel studied the single trait. Sagret studied the inheritance of the contrasting traits, and the other scientists were studying the whole organism, which was actually the not possible to draw the results like the Mendel. Um, the as well as the approach of the Mendel is concerned, uh, with the approach, scientific approach of the Mendel, there are many things which supported the Mendel. I always say to my student that Mendel was a lucky man. Uh, to somehow, I think that God gifted him his luck as a you can say as a gift behind as a gift for those hard work behind his experiments. The good luck of the Mendel was that he studied almost around 7 traits on the Pisum plant, and none of the trait was on this single chromosome. None of the trait was all different. All the traits were scattered on the different chromosomes. Say, for example, he studied the 7 traits. 7 traits were on the 7 different chromosomes of the Pisum stigma. So the procedure of linkage was not the hurdle in the Mendel's finding. If any trait based on the single chromosome, if any trait if based on the single chromosome, such kind of result cannot be obtained as Mendel has obtained these results. In 1900, after 1866, up to the 1900, Mendel work was hidden. At Mendel work was hidden. None of the scientists focused on the Mendel's work. In 1900, Mendel's paper in 1900, 1900, 1900, Mendel's Mendel's paper was rediscovered, and there were 3 scientists who rediscovered the Mendel's paper. The one was all of these were the botanists. These were the botanists. One was the Hugo de Veris. Hugo de Veris. Hugo de Veris. He was from the Holland. Uh, this Hugo de Veris was known for his mutation theories and studied on the evening primrose and the maize. The next scientist was the Carol Corin, C-A-R, Carol, Carol Corin. He was in the Germany. He was a German scientist. He was the German scientist and working on the maize, peas, and the beans. Next one was the Eric von Sissinich. Sissinich. He was in the Australia and worked with the several plants, including the garden peas. Each of these investigations obtained evidence for the Mendelian experiments. Each of these supported the Mendel experiment. After their work, the Mendel experiment was reborn. In addition to the naming the science, Batson actively promoted the Mendel's view of paired gene. Batson actually promoted the Batson Batson promoted the Mendel's theory of paired gene. He used the allelomorph, subsequently shortened to allele, to identify the members of the pair that control different alternative traits. Uh, Also, during the early 19th century, French scientist Lucien Cuno showed that the genes controlled fur color in the mouse, and American W. E. Cassell related the genes to the sex and to the fur color and pattern in the mammals. Later on, Dane W. L. Johnson studied the influence of heredity and environment in the plants. Johnson began using the word gene from the last syllabus of the Darwin's term pangene. Batson used the term allele for the actually the term was the allelomorph, which was shortened to the allele. Darwin used the term pangene, which was shortened to the gene. These pangene and the Mendel's factor was almost the same word. Later on, William Rocks. Later on, the William Rocks. Okay, I told you that Darwin used the term pangene, which was shortened to gene. It was shortened to gene. Next was the William Rocks. William Rocks, he postulated as early as 1883 that chromosomes with the nucleus of the cell are actually the bearer of the heredity factor. He told that the Mendel explained that there are some paired factors which are controlling the hereditary or which are controlling the transfer of the trait from one generation to another generation. He told that those controlling factors are actually present on the chromosomes. The only model he was able to advise that would account for his observed genetic result was a row of lined up objects duplicated exactly. After all these working, uh, English physician in 1900, an English physician, Sir Archibald Gear was working on the biochemical pathways. He was actually studying the congenital metabolic disease in the human. On one of these was the alkaptonuria, which is caused by the block of the catabolism of the amino acid phenylalanine and tyrosine. Geert proposed that alkaptonuria was due to the single defective gene that produced a non-functional product resulting in the metabolic block. At that time, after this discovery, Geert results and his interpretations of these results was described in detail of his book. Geert Geert actually wrote a book, Inborn Errors of Metabolism. Inborn Errors of Metabolism. He used the term one mutant gene, one metabolic block. One mutant gene, one metabolic block. One metabolic block. Like the Mendel, this theory was also ignored by the scientific community until 1941. Both the works, the Mendelian work and Archibald Geer's work, was ignored for at least 40 years, 40 up to 40 years, yes. After 1941, Beadle and Tatum rediscovered his concept during their neoclassical work on the neurospora, and they refined his theory from one mutant gene to one metablock to one gene, one enzyme defect. They said that one gene is responsible to create one enzyme. Subsequently, this concept was revised to the more accurate concept, and now we people know that one gene is actually responsible to control one polypeptide. Now this concept is refined to one gene, one gene, one polypeptide, one polypeptide. This is all about the history of the genetics. After that, after this, the modern genetics was born. Now we are working on the advanced techniques of the genetics. This is all about the basics, or you can see the history of the genetics. Thank you, students. Hoping it will be easier for you to understand. Thank you. Allah Hafiz.